Screen to go live. If somebody could confirm that that is coming through well, that would be helpful.
Done. It's going live on YouTube.
Yes.
It is, because I heard myself saying that. And this is our fourth. Fourth.
Yeah.
Okay, I think we can start. It's 1100 hours in Eastern US. You know what time it is, wherever it is that you are. We welcome you all, and thank you for joining us in our fourth talk of the Frontiers in Social Evolution seminar series. It gives me great pleasure to introduce our speaker today, Aaron Sandel. Aaron is an assistant professor at the University of Texas in Austin in the Department of Anthropology. And before that, he completed his PhD at the University of Michigan, working with John Mitani. His research, while he was doing his dissertation, focused on examining behavioral and hormonal correlates of the transition from adolescence to adulthood in male chimps. I'm quite sure I first met Aaron in 2015 or 16 when our colleague Charlie Nunn invited us to be part of a workshop that he organized to discuss of issues of data quality. I'll also admit that when I met him, I had not read the 2014 paper that he published with Patterson and other colleagues discussing issues of data quality on group size, primate group size. And what an eye-opener, right? Because we would, I mean, if we were given a list of things that we should be able to more or less estimate and report and analyze without lots of issues, would be something like group size, right? The first reaction is, well, it cannot get any easier than just going and counting animals. But I'm talking to an audience that fully appreciates that even trying to get proper data, high quality data on group size is quite some challenging. So after finishing his PhD, he has continued working in GOGO on chimps, but he's also become much more interested and much more committed to issues of the science around comparative studies with a focus on data quality. And I was rereading the papers that he suggested we have a look at. And here's a young scholar telling us in the introduction, this is not the concluding sentences in the discussion where we try to throw something there about how this is going to be important for conservation or we need more data. We were unable to draw statistical conclusions about the evolutionary pressures leading to the Bruce effect. That's the ending sentence of the introduction. So, and then for the analysis, for the methods, we did not proceed with the formal phylogenetic comparative analysis. We did not proceed with the formal phylogenetic analysis. We chose instead to limit our analysis to solid, thorough, descriptive data analysis, which I think has been a bit of a theme around here that we need both. Of course, we're not saying one or the other, but it gives me a lot of pleasure to welcome Aaron to tell us the way he's been thinking about these issues with regards to the study of social evolution, namely on chimps, but also more broadly across various taxa. Before I give Aaron control of the talk, let me just remind you of a couple of rules. He'll probably be talking for anywhere between 40, 50 minutes, I assume. I ask that we please mute yourself, that we're going to have no questions during the presentation. The questions are going to come after, during our Q&A session, question and answer session. It's been fantastic. We have stayed with some of you up until 2 p.m. talking about all kinds of things with with people who have already a career well established, with young scholars, young students. It's been great. We encourage you to join us. During the question and answer session, I'll repeat it, but we ask that you just indicate your willingness to pose a question by just typing a question mark in the chatting window. This is something personal, but I really discourage chatting. I don't really see the difference between 2 or 3 of us chatting between us. It's not very different from like if we're talking to each other when we have a colleague talking. So I invite you to reflect on doing that while somebody's talking to us. I think that I need to make sure that Aaron, you are in charge by sharing. Do you want to share your screen with me?
Sure.
And with that, unless Lauren or Karsten, please help me if I am forgetting something that needs to be said. If not, we welcome Professor Aaron Sandel from University of Texas, Austin. Aaron, take it from here, please.
Thanks, Eduardo, for the great introduction. Let me just get this screen sharing up and going.
Can somebody confirm we're live and okay? Okay, thank you.
Yeah, so thanks again, Eduardo, for the introduction. Thanks to Lauren and Karsten and Eduardo for organizing the series. It's been a lot of fun attending. And thanks, Eduardo, you mentioned maybe it being impressive that I said that I didn't proceed to do an analysis at the end of an introduction to a paper. Maybe there are others in the audience who with more statistical savvy would have figured out a better way to do that. But that's one of the things I want to talk about today. But as you mentioned, the focus of my research is on chimpanzee social behavior. Here's a male chimpanzee grooming, moving his hands through the back hairs of another adult male chimpanzee, a classic scene in the social lives of chimpanzees. But today I want to sort of zoom out from my work studying this one population of chimpanzees at Ngogo to reflect more broadly on the field, to discuss within species variation, data quality, and the aims of primatology. And that sounds a little grand, but it follows some of the themes highlighted by Karsten, Lauren, and Eduardo in this series, a lot of the people who are in the audience today. And again, I really appreciate being invited to talk today and participate in this series. And I'm excited, especially to have the discussion afterward. To start, I want to sort of tell you how I got interested in some of these questions of data quality and within species variation in comparative studies. And this is, it started with a side interest of mine, sort of one of my hobbies, which is the evolution of human hair. And this is, maybe I should get more hobbies if that's what I'm saying is my hobby. But I'm interested in human hair evolution. So I did a comparative study several years ago, going through the published literature, combing through the literature, finding as many studies I could on hair density in as many mammals as possible. And I'm not going to go into the details of this study, but one of the conclusions that I took away was primates compared to many other terrestrial mammals are sort of less hairy. They have fewer hair density, less hair density than other mammals. And chimpanzees are one of the least hairy of the primates and also less hairy than a lot of other mammals. So I thought this was relevant to telling the story of human hair evolution, that humans are on this spectrum of hair density with other apes and other primates even. So that's all well and good, a little study on hair density in mammals, but there were several problems with this comparative study. First of all, there were often small sample sizes. The chimpanzee data that I reported in this comparative study relied on a sample size of 1. I was going through this great study that Schultz did in 1931, and he had reported hair density in a few different chimpanzees, but I limited it to the one adult female who didn't seem to have any health issues. And actually a lot of the primates and many other mammals in the study relied on only one individual. So that could be a problem. That might not be capturing the true value for some of these species. Another problem is that for some taxa, I had very few species represented. For example, only 5 rodent species. Related to this, there were sort of some species that were missing entirely that especially could be relevant, like, for example, elephants, which are very large, pretty hairless. There's also the issue of measurement error. When I'm combining data from different sources, some hair density came from animals that had been recently killed, other ones had been preserved. Occasionally it was from live animals taking sort of sections out of their hair. So with different methods, different ways of measuring hair density, there could be just error in those methods, making them less comparable. Another problem which really interested me, but I didn't really know how to deal with it, was within species variation, true within species variation. For example, seasonal variation. For a lot of these mammals, they had very different hair densities in the winter and in the summer. But for the purposes of this comparative analysis, I just took an average, I just took the mean. So I was compressing at times huge variation in hair density within the same species, maybe even the same population, into a single data point to get a mean value. And I sort of described my methods to the best of my ability in that paper, but sometimes there wasn't always transparency in what I did. For example, in a study of macaques, rather than taking a square centimeter of skin and counting the hairs there, they were taking a 1 centimeter circle. So, you know, on a piece of paper, I was on the back of the paper of that article, I was calculating the area, you know, density per area to make it comparable to the other studies that were maybe taking a square centimeter. So there wasn't, for a reader, they could look at my supplementary material, see the results that I was presenting, but they wouldn't have seen that transformation. Another problem with this study, especially as it related to the question of human hair evolution, was I didn't plot humans. And this is partly because many studies of human hair density, they're counting visible hairs, and many studies report no back hair density, no visible hairs in the back. And I was working on this study around 2010, 2012. I knew this wasn't true for all humans. I will say, you know, I've been interested in hair density for a long time. As I think more and more about hair density, I've also become more and more hairy in some areas, well, less than others. But I knew that humans have hair density, but it actually is sort of their methodological problems in counting hair density. There was one sort of descriptive study in the early 60s. There was somebody who was studying hair density in human populations. He reported that there is variation in hair density in humans. He didn't count the hair densities though. He actually had a method that probably wouldn't be approved these days. He went into the locker room of a gym, and with a little note card watched and sort of sketched people's hair densities without their knowing, and then in doing a little small talk would get their age for his analysis, which is actually very similar to a study, to an episode of Seinfeld where Kramer's trying to learn methods of how to best shower. Anyway, I know there's hair density variation in humans. So when I was presenting this at a conference in 2012, I plotted one sample of one individual that I knew the density of. And I wasn't that transparent about it, but it was me. But this is another point, is that within species variation, you see changes in time. So for example, if you even looked at the same human, an adult male several years later, you may see a dramatic difference in hair density. So there are these problems with this comparative study of hair density, but I realized that these are problems that you find in a lot of comparative studies. And this becomes even more difficult when you're dealing with more ambiguous and variable traits like social.
I know.
So I want to shift focus to primate social behavior and then sort of zoom out again. What are the aims of primatology? And I'm sort of thinking about this from a contemporary perspective as still being a young scholar in the field, but also looking back at the history. For example, there's this one nice quote: "In the attempt to reconstruct the life of man as it was shaped through the ages, many studies of primate behavior are now underway in the laboratory and the field." This is this focus on studying primates to sort of understand human evolution. Also, early on, there was another focus on studying primates across species to understand general biological evolutionary processes. This study by Crook and Gartland, they're quoted as saying, "Our purpose is to direct attention to patterns of relationships inherent in the data now available and to begin the task of ordering it." So I see these sort of two themes falling into one aim, which is to infer evolutionary processes by comparing species. And oftentimes there's a special focus on human evolution, but not necessarily. And then the other aim is just studying the details of one population of primates, the individuals, the group of primates living in a specific location. And these aims go hand in hand. For example, in order to make the comparisons across species, you need the detailed observations of populations, the detailed observations of the data on which the comparisons are based. And they also sort of reflect disciplinary differences. The comparative approach is more the focus, the starting point maybe for behavioral ecology, whereas studying the detailed descriptive studies of one population or one species is more of an ethological approach. They go hand in hand. There's sometimes little tension, but I also want to mention that especially this comparative approach, it's also grounded in completely different disciplines like anthropology, psychology, as well as other aspects of biology. And because of these multidisciplinary sort of origins to primatology, you also get different methods. So to some extent, studying the details of one population draws on some ethnographic methods, almost. And it usually makes sense to first study populations in detail and then zoom out and make comparisons across species. But over the 70 years of field studies of primates, it's also possible to sort of sit back and just focus on the comparisons with the available data in the literature. And there have been huge developments on both of these aims. And some of the most notable studies, I think, especially those recently, are ones that compare across a wide range of species to infer the adaptive value of certain traits and to understand some of these evolutionary processes. And these are really exciting. These are the theories of our field, or at least the major hypotheses that get, I think, a lot of researchers really excited and want them and excited to go out and test a hypothesis, try to refute a hypothesis. And there are also ways to guide how we sort of see patterns in nature. But there's a concern that there may be too much of a focus on comparative analyses and not enough on descriptive studies. Here's a quote: "We must hope that the descriptive phase is not going to come to a premature ending. Already there are signs that we're moving into an analytical phase in which the ratio between experimental analysis and description is rapidly increasing." And this is a quote by Tinbergen in his 1963 paper describing sort of the aims of ethology. And I think his concern is all the more urgent now, nearly 60 years later. And this is especially true when comparative analyses come to conflicting conclusions. And I think we see this replication crisis in a lot of disciplines in biomedicine, in psychology. To some extent, there are problems of replication also in primatology, and it may be worth pausing to pay careful attention to the data that these large-scale comparative studies are based on. So there's several problems with the comparative studies that I identified and I sort of encountered in my study of hair density. Another problem I should add to that list is sometimes erroneous data can enter data sets. And going back to what Eduardo said when we first met, which was actually 2014 at this working group on data quality in comparative studies, a large group of us got together, Carola Boris, Charlie Nunn were organizing this, and we were discussing, okay, what are the issues with data quality in these comparative studies? And one of our sort of framing, the way we framed this issue was that recent decades have seen rapid development of new analytical methods to investigate patterns of interspecific variation. Yet these cutting edge statistical analyses often rely on data of questionable origin, varying accuracy, and weak comparability, which seem to have reduced the reproducibility of studies. It's time to improve the transparency of comparative data while also making these improved data more widely available. And in that working group, in the subsequent paper that we wrote, we identified several principles for describing the data, including metadata, being very transparent in your procedures, and making sure that readers can interact with your data. And I want to discuss these problems and several other sort of principles that we may have when conducting high quality comparative analyses. And then at the end, I want to discuss sort of recommendations about connecting this to how we should be doing primatology, which I'll argue is a return to the descriptive phase. And I want to use the example of group size in primates. And to start with a hypothesis that has guided a lot of my research interest, the social brain hypothesis, which was has been sort of first and most often discussed by Robin Dunbar. And there's sort of two versions of the social brain hypothesis, I think. One is that species living in large groups require more complex cognition and thus evolve larger brains, for example, larger neocortex, or sort of a related but flipped sort of evolutionary, the causality is different, which is that brain size, for example, the neocortex, limits social group size. Both of these would predict a positive correlation between a species' typical group size and their brain size. And you know, there seems to be really strong evidence for this relationship, but a lot of the published studies looking at this rely on one data set of brains from Stefan et al. They went through and did probably a good, I'm not a neuroscientist or an anatomist, but they seem to have done a good job cutting open some brains. But it was ambiguous from these studies of the social brain hypothesis exactly where the group size data were always coming from. And so just to sort of reflect on this, Sam Patterson, Jordan Miller, John Matani, and I got together and we sort of questioned how our data collected, taking the example of primate group size, and how should they be collected. So we came up with sort of strict inclusion criteria for what data, if we're trying to really determine what is certain species of primates, what's their typical group size, how are we going to determine this? Our inclusion criteria included only getting data from wild habituated groups, making sure that all the individuals were known, or at least the age, sex, classes of all the individuals, so you're really sure that you've counted everyone in the group. Relying only on observational studies, not just estimating from surveys or a census. Only including data where there is an exact count of the group size. Sometimes studies were publishing a range or an average. only including studies that had a minimal populations where there was a minimal human interference. So not if they're being actively provisioned, for example. And then we wanted to make sure that there was a reference for each datum, for each data point, a reference from the primary literature. And just really briefly, we compared this to several other data sets of primate group size, and many data sets don't report how they included the data that they did. They weren't really reporting whether the primates were habituated, or whether all the individuals were known, or whether they were relying on group size counts from a survey or an observational study. So they just weren't really reporting these data. But we found for many of these studies, including comparing our study to that of the original Dunbar study, that the group size values were comparable. They were similar, although there was some variation with other data sets when we were looking at our sort of high quality data set versus other data sets which could have been high quality, but they weren't really reporting how they were getting those data. And so 5 out of the 7 we compared were pretty similar, although many of the studies seem to undercount the number of individuals in the group compared to our study. So the first question is, how are you including data? How are you collecting data? Another sort of related issue, which again was a point that I noticed in the study of hair density, is this idea of within species variation in group size. We've known from the earliest studies of primates that primates vary a lot in group size. So even if the data may be high quality, another huge issue in doing comparative methods is, how do you account for within species variation? And this has been a concern of several primatologists for a long time. Tom Strusaker wrote that until this level of variation, this within species variation, is understood and taken into consideration, broad interspecific comparisons and generalizations are misleading, if not counterproductive, in furthering the field of behavioral ecology. Similarly, Karen Stryer wrote, the compression of intraspecific variation in any behavioral trait into a single species-specific value precludes interspecific comparisons. So they were identifying that, wait, primates are variable. We can't just go and taking an average value, like a mean for something like group size or another behavioral trait, and then start comparing across species until you really quantify and maybe account for that variation. So this is what we did with our data set of group size. We wanted to see to what extent did variation group size influence this social brain hypothesis, this positive relationship between neocortex ratio and group size that's been pretty well supported in the literature from this data set. So we had all these values for group size. On this figure on the right shows the black dots are the mean value per species, and the open gray circles are each different populations, different group sizes. So there's considerable variation in group size, and with this data set, looking across these 23 species, about 70% of the variation in group size was due to between species differences, which means, the flip side, 30% of the variation in group size was occurring within species. So there is still, overall, species did seem to differ, in group size, but some were there's a lot of variation within species. The problem then was, how do you incorporate that into a comparative study, which often relies on just a single value? If you're comparing species, it relies on a single value. How do you include that within species variation? We applied a method that had been developed around 15 years ago to incorporate within species variation into a comparative method by treating that within species variation as sort of measurement error. So we included this standard error of group size for each species as error in the model. I'm happy to talk more about the details in our discussion at the end. I just want to sort of go through the broad strokes of this. What it does is it will just essentially shift the slope of the regression to account for species-specific standard errors. And here's a figure showing the black line is without taking into account within species variation. And then the dashed line is when we included within species variance standard error as an error term. The black is, the black is for all the species in our study, and the gray is when we were excluding pair bonded species that only seem to form groups of they're two individuals plus their offspring. And controlling for standard error group size per species, controlling for within species variation group size did not really affect the results. We still recovered this positive relationship between neocortex ratio and mean group size. And again, I'd be happy to talk more about the statistics at the end. By controlling for within species variation, I don't think this is exactly capturing what we wanted to do, because it's not an error term. It's actually, it could be potentially biologically meaningful variation. So we did another, so yeah, sorry, that's the pair living species, group living species. We did another method to sort of more describe this variation, especially more visually. And for this, we did a resampling procedure where we just took one value of group size per species and sort of treated that as the species typical value. So what if we just go through, we say we only did a study of one population per species, and we thought that that was the group size for that species. And then we run a regression. Do we recover a positive relationship? And then we do that again, randomly choosing one value per species. Do we recover this positive relationship? And if you repeat this enough times, like 10,000 times, we wanted to see whether overall you tend to get this positive relationship, somehow to account, sort of to visualize the certainty of this relationship. And we found that we do recover this positive effect of neocortex ratio and group size. There you see this variation, but it still seems to support this positive relationship between neocortex ratio and group size, even if you're taking sort of different populations in terms of the group size. We repeated this by excluding pair bonded species, partly because their mating system and social system may put certain constraints on group size. And excluding these pair bonded primates, again, there still was a positive effect, but there was a lot more variation. You know, sometimes, occasionally, you would even see sort of no relationship between neocortex ratio and group size, depending on what random group size values you were selecting. But this is also a case where we, established that about 70% of the variation was between species. So there were differences in group size between species. This method would probably be especially important. Both methods of controlling for within species variation and visualizing it are important for traits that would be even more variable within species. And just a quick follow-up on this study, if you look at different species and use some different methods, you may find different results. We just quickly, we had these group size values for primates, and then we found another data set of brain sizes. This time it wasn't the Stefan et al. Study, it was data from MRI, from Rilling and Insel on brain sizes in a smaller number of primates. And we looked at within species variation in group size and looked at this related to neocortex size. And here there wasn't really a strong relationship between neocortex size and group size, even just looking at average values. And I think this is partly because it's the species under study. This data set included orangutans, which have large brains, but live in small groups. And I haven't sort of gone through these data carefully, and there have been a lot of other people who have since been publishing more on this effect of neocortex size or brain size and group size. And again, I'm happy to talk about this more, but this sort of raised the question of species under consideration. Is really important. So from these studies of group size, I first of all got a little burnt out. I was like, okay, there are a lot of problems in comparative studies, including in primates. I was working with an undergrad student at Michigan who wanted to do a comparative study of the Bruce effect in rodents, this phenomenon where a female will enter pregnancy, terminate her pregnancy ends when a new male is introduced. And we wanted to do a high quality comparative study, taking all these principles that we had been thinking about. And what a perfect group of animals to look at, rodents, because there are a lot of rodents. This is where the Bruce effect has been studied. We know so much about their reproductive physiology. We know a lot about their behavior. So if we're going to do a good study, a good comparative study of reproductive patterns, let's do it in rodents. Also, I like, as Carson said, I think in his talk, something to the effect of most mammals are mice. I've said that before. So we looked through the literature on the Bruce effect in rodents and wanted to test several hypotheses for why the Bruce effect occurred in rodents. One being as potential a way to avoid infanticide when a new male is introduced. Maybe it's adaptive for the pregnancy to end so that the female doesn't start investing more in this infant that is maybe doomed. It might die from an infanticidal male. Alternatively, maybe the female, the pregnancy is terminated because if a new male enters, that might be reflecting the fact that the old male, the biological father, has left. So we wanted to look at the Bruce effect in rodents. We wanted to look at evidence of infanticide and evidence of paternal care. So we go through the literature. This is with Rachel Stokes, who was an undergrad at Michigan when she started this project. And we looked through the literature. This was a lot of her work. And we found data on the Bruce effect. And we found some data on infanticide and paternal care. And we reported it in sort of the tables and tried to make sure each data point that we're referencing had that we reference every data point. And here's just a figure looking at our results. The Bruce effect is this column on the left. Black means that they have a Bruce, that they exhibit a Bruce effect. Gray is that they're variable. White is that there's evidence they don't have a Bruce effect. And then we looked at this in infanticide, paternal care. Do they have infanticide? Is it variable within that species, or is there evidence that there's not infanticide? Same with paternal care. And just looking at this descriptively, this is what Eduardo was mentioning in his introduction. We see some variation in the Bruce effect, but there wasn't really enough variation in infanticide, and especially not in paternal care, to be able to do a correlation, really, I think. And again, this is where maybe someone with more statistical savvy than I could say, oh wait, you could do this in a more creative way. but you're just looking at the data. There's not, you need variation in both traits to be able to model whether one is predicting the other, or just to model whether there's a correlation. So we decided to sort of terminate our goal of testing the hypothesis for the Bruce effect because of this missing data, because there was a lack of actual negative results. And instead, we just published this huge annotated bibliography as a supplement to this more descriptive paper, and a descriptive paper highlighting some of these problems in comparative methods. And this was, again, Rachel Stokes just did this huge annotated bibliography where we're reporting the methods that were in the study and how we were classifying these various behaviors. And this way, if someone is going to be studying the Bruce effect in the future, they can look back to our paper and use that sort of as a jumping off point to do a comparative study. And this also connects to something that Carsten raised in his talk a few weeks ago, which is that a bulk of what we were doing is sort of adding, as scientists, we're trying to add to this collective data set, and that we can and we should test hypotheses along the way. But to some extent, maybe our legacy, as Carsten was saying, are maybe the data, not the hypotheses. Okay, so just to pause to review briefly, there are several problems with comparative studies. There are some ways to mitigate these problems, like having inclusion criteria, having metadata, including multiple data points per species, assessing and incorporating within species variation. And I think in addition to mitigating these problems in comparative studies, we also need to return more to a natural history phase, a descriptive phase. Here's another quote from Tinbergen in the same page of that same 1963 paper. The variety of behaviors found in the animal kingdom is so vast, and their description is so much more laborious than the description of structure, that selectiveness of description will become increasingly urgent. This will only be possible by a more explicit formulation of the problems we wish to study, and by growing certainty about the nature of the data we need. So this is some sort of an idea of a informed, targeted natural history phase. And I think this is important for several reasons. We can do this targeted natural history to identify where the holes are in the studies. For example, with the Bruce effect, there weren't enough data on infanticide and paternal care, at least as it related to the Bruce effect, to really robustly test that analytically. Also, I think a return to observation is important because with animal behavior, in many respects, it's a qualitative science. The actual study of behavior is qualitative. We were trying to make various motions and things that animals are doing into these. We define them, for example, even grooming. There's this process where we're saying, okay, this is grooming, and I'm going to say this in my ethogram and give it this operational definition, and then we can start quantifying it, and we use quantitative analyses. But the process of developing that ethogram is a qualitative method. Related to this, the human observer in animal behavior is the apparatus. Whereas in, say, physiological genetic studies, I mean, humans are really important as experimenters and technicians, but to some extent, the pipette is the apparatus. The pipette is what's measuring out the values of the liquid, or it's the plate reader is the apparatus. Whereas in animal behavior, it's us. We're watching them. There's so much that is filtered from when we're watching to recording what we're seeing. So it's sort of that the human needs to be calibrated. So in thinking about the future of primatology, I think it's important that we have a return to emphasizing observation, description. Related to this, I think it's important that we think about who's collecting data. On the one hand, if we're going to be studying animal behavior, I think if we can, we need to spend as much time still keep that foot in that door. If we're studying animal behavior, we should be watching animals behave. Of course, we could be collaborating with others who are also collecting data. But also, I think we need to expand who we're including in our field as scientists. In primatology, at least my experience with chimpanzees, a lot of data collection is done by field staff who are living in near where the chimpanzees are studied. I think this is true across many primate studies. And I think it's important that not only are those people just research assistants collecting data for us, but that we're training them and mentoring them to be collaborators. And I think they're gonna, having a wider range of people watching the animals is gonna really be important for gaining new insights and taking a more holistic approach. And just, I wanna give one case study to illustrate this point. Case study of some research that I've done in Kibale Forest in Uganda. Here's just a video of Kibale Forest at Ngogo, sort of in the middle of the Ngogo territory, which is in the middle of Kibale Forest. This is sort of, you know, the sort of vibe that you get among ecologists is that it's sort of a pristine area. This wasn't an area that was logged in recent times. It was maintained as a nature reserve. So this is, you know, a beautiful classic just photo of Ngogo. But what do you notice in this photo? Is there anything that stands out to you? And I don't need to call on the audience, but there is a rock in the lower right-hand screen. And this just looks, I mean, it's a naturally occurring rock, but it is sort of flat and dented on the top. And it turns out that this is a grinding stone. And there are grinding stones that are scattered throughout Ngogo. And this is, I mean, researchers have sort of been aware of this, but I think that sort of the first people to ever mention these have been Ugandan scientists. And when I first came to Ngogo as a graduate student, it was the Ugandan field assistants who pointed out, oh, there's a grinding stone. And I, you know, throughout the years, people would say, oh, there's a grinding stone, here's a grinding stone. But I was thinking, and this is what other Ugandan scholars and other primatologists at Ngogo discussing this, if there are grinding stones scattered around Ngogo, this would indicate that there was a historic presence of humans, maybe a widespread historic presence of humans. So a couple of years ago, with several colleagues, we sort of collated all the grinding stones we could think of, and I was doing more of a targeted, you know, keeping my eyes open for these grinding stones. And we found that there were scattered within the Ngogo chimpanzee territory, there were around 20 grinding stones, sometimes within a few hundred meters of each other, sometimes even closer within 10 meters, other times separated by several kilometers. So this indicates that there was sort of a network of maybe small scale villages throughout Ngogo in some historic time, it may be as recently as 100 years ago, potentially much longer. And this could explain some of the habitat heterogeneity in Kibale, which could then in turn explain some of the patterns in the demographics of primate groups and different, the structure of the animal communities in this forest. And just to sort of start addressing this, we did little ecological plots around each grinding stone. And these are areas of the forest that don't really seem disturbed, but we found that, for example, with basal area of trees, that areas right around the grinding stone had a basal area that was intermediate between what would be like a heavily logged area and a lightly logged area. So this is just to illustrate the point that taking this holistic approach, including one that relies on Ugandan scientists and Ugandan field assistants to sort of, they bring about new questions that could give insight into basic patterns of ecology and primate densities. this study that we did was with two of my co-authors were Godfrey Mbabazi and James Tibissimwa. Godfrey is one of the field assistants who collects data on the chimpanzees. Big James Tibissimwa is the head of the snare removal team, but Tibissimwa, and also doesn't speak English, but he has this sort of expert knowledge of the history of the area through oral history and from growing up as a hunter. And I just want to show a video of the co-authors. We're all in the forest and sort of asking some questions more about grinding stones. And Godfrey is asking, to what extent were there really people at Ngogo?
That many people were in Ngogo.
No, but I mean,
and then Godfrey was explaining that Big James was saying there were many people at Ngogo.
Is there a place where there are many grinding stones, where you find many grinding stones?
Ngogo wagamba aliho akachweka kama na mbele abantu bengi atuso kujya ubundi tukasera aho tembengo nyingi.
No, I think I want to
But it's these conversations that sort of can give insight into broader patterns of ecology and primate populations.
That there were very few people and that other people were in Kisen side, others in Kitokoma side, so they were just scattered.
So thinking about the future of primatology, I think that we can do this return to observation, think about who's collecting the data, broadening who we're treating as scientists and who we're training as scientists, and taking this holistic approach. I think this is sort of, it's also advice for me as I'm continuing on my career studying primate behavior. But I think this return to a descriptive phase will also lead to new insights and new hypotheses. Thank you.
Thank you, Aaron. Thank you all for joining us and listening to Aaron's presentation. For those of you who may not have attended previous talks or who were not able to join us from the very beginning when I made a couple of basic announcements. So we now move on to the question and answer session, and we are going to have that for about an hour. So during that session, I will be monitoring the chatting window. I ask that you do not write a question, just type a question mark. And it's worked for me in the past, I just keep a list of the names of people coming in with questions, and I just go in the order that you post your question mark and call your name, and that way we can have an exciting back and forth with Aaron. Aaron, can you stop? I don't remember why, but I need to ask you to stop. So you don't have to share your screen now.
Am I still sharing?
So turn off your screen sharing.
Is it on or?
I don't know. No, I don't think it is. So it's always, again, we would like to remind you, I think it helps if we just stay muted unless you are asking a question, in which case I will be calling your name. And you know how it is, sometimes it may take me a little bit to locate you visually. Most of you have your photographs, but I'll be calling your name. So thank you so much, Aaron. And we can now take the first question for Aaron. Just send me, type your, if you have a question. Kaya Tombak. Kaya, where are you, Kaya? There you are. Kaya, please.
Thanks for the talk. I was just thinking about the appropriate unit of analysis for comparative studies. And if your question, let's take the social brain hypothesis, for example. If your question is about whether brain size is a limiter of group size, I wonder if the maximum group size is more what you would want. And within species variation is still important, but more in that maybe the number of populations with data could just increase the certainty that you have in the maximum group size value. And so, but then if your question or if your hypothesis is more that brain size is a facilitator of increased group size, perhaps some kind of measure of central tendency is then the more appropriate value. Does, what do you think about fine-tuning the measure that you use from these data sets and how much we can improve our inference from comparative studies by targeting our data analyses that way?
I think that's a great point. And to some extent, you don't need to be looking at a mean value and then somehow incorporate the variation. You could be, I think the point about looking at maximum group size could be really relevant to a certain question. So it's still you would still want to get an accurate value of maximized group size, maximum group size. So looking at enough populations, maybe look at variation within a population and see what's their maximum group size, and then do that across populations. This requires long-term study. But I also think that depending on the question, for example, variation itself could be relevant. So another way to look at it would be to model something like brain size against variation in group size. You may have sort of different predictions. And this is, once you have the key thing is having I think the data set with multiple values per species, and then you can sort of choose. So when you're publishing data sets or compiling data sets, making sure that those data are available. Another thing that Laszlo Garam-Szegedy and some of his colleagues have done is sort of taking the median and then maximum, minimum in some of their analyses with other non-primate animals.
Thank you. I will ask if you can please, we have a wonderful eclectic community. I know some of you, I don't know everyone. So if you very quickly just introduce yourself, saying where you are, and then that way we get to know each other a little bit more. The next question comes from Sari Van Bell. Sari.
Hi, Aaron. Thank you very much for this interesting talk. I have kind of a common question. I don't know yet where I'm going to the last part of your talk in proposing that who we consider as the scientists, the persons collecting the data, and they are researchers that based in the northern part of the world who are generally assumed to be like these scientists that then have persons collecting the data for them, and certain researchers have personnel that do that over the course of a long career, and then they have others that use students that rotate through a whole bunch of people collecting data. It's my own experience, the more I am collecting data, the better I get at the details of collecting the data for the specific scientific question at hand. So there should be variation in the quality of data collected according to the strategy of the researcher. And I don't know if you want to kind of give an opinion of that. The other section would be who we consider as the researchers and how that help those persons that collect the data. How do they benefit in their career? Because we publish and that's how we benefit and we get promoted and we get grants. But how can they get better or promoted in their career as a data collector?
Those are great points. Thanks, Sari. Yeah, I think first of all, I want to acknowledge that people who are doing long-term studies, of course, have to rely on people who are in the field all the time. So that could be field staff from that primate habitat country, which is a great way to bring jobs to areas, especially, for example, in tropical forests. They're often surrounded by rural areas where there could be high unemployment. And then there's this other model you're bringing up of having these more short-term researchers. I think that there's nothing inherently good or bad about either of those. There is something I think that we should be aware of, especially for European and American, United States researchers going to a place like sub-Saharan Africa and doing research, we have to be careful that we're not accidentally sort of playing out this colonial dynamic. But I think that that can be done through really sincere collaborations and just good management and good teamwork. But I think your second point about, and that sort of relates to your second point about how the data collectors can benefit, I think that it's probably useful to have paths toward becoming a scientist if they want to. I mean, in some countries, maybe there isn't as many career options, but then, I mean, why not create, like we could sort of create them. You could have a supply and demand, or the supply could create a demand. If we were training, for example, I work in Uganda, we have a whole new group of Ugandan field assistants who have been hired over the last few years. They're all seem really motivated and excited to watch the chimps, but some of them want to pursue higher education. So how can we as primatologists, and especially those of us who are working at research universities in places like the US, Europe, support them in their goals of higher ed? So there could be, maybe they just want to hire, maybe they just want a university degree, come back to collect data and become a data collector that for a career, but then when they retire, it might be nice to have that bachelor's degree, something like that. Is that getting sort of, those are sort of first things that come to mind.
And this is rather also an open question. It's not that you need to solve this issue, but it's definitely something that we should consider. How can we help local data collectors to become scientists and actually participate in the scientific community? And then obviously we also have to talk about the language, and that you presented us this video of this one man who seems to have ample information, historical information, but does not necessarily have the language that scientists use, and that is not his fault. It's just that this is how it is.
Yeah.
Anyway.
Yeah, I think, I mean, right, it's not, I'm not trying to say that I have the answers, I definitely don't, but I do think it's all of our responsibilities to be brainstorming those answers. And I attended this African Primatological Society meeting a year ago in Entebbe in Uganda, and there were some great discussions there about this exact issue. Joe Setchell was saying maybe we should stop calling field assistants, treating them more as collaborators, things like this. And also I think there is an importance to making sure that we train the data collectors who are from the primate habitat countries to become scientists if they want to, make sure they have that option, because I think the future of primatology should be focused more on scientists in the countries where primates are. And then so it's a real collaboration rather than just this sort of, I mean, we don't want to be just mentors. I want to be mentored. And someone like James Tibisimwa, who doesn't speak English, he has a vast knowledge of the through oral history of the area. He's someone, for example, that I would want, he should be a adjunct faculty at a university teaching conservation education in Rutoro, in the language in Western Uganda, because I mean, I certainly take that class. I first need to learn Rutoro, which is bad that I haven't yet. Now I have it on film that I need to learn Rutoro. I have my flashcards around the corner. Sorry.
All right, thank you.
So we have many questions coming up. I just run quickly the names so that you know that, yes, your time is coming. We have Filippo, Joshua, myself, Karen, questions from YouTube, and Suleima. So that you know that you are on the list, it's happening. Filippo, you're next.
Thank you, Eduardo, and thanks, Aaron, for the nice talk. My name is Filippo Aureli. I'm from Universidad Veracruzana in Mexico. And I want to comment briefly on the discussion you had with Sari, and that I think it is really important, your point, the giving something, but my feeling is that giving something they want, the local people want. and not necessarily everybody wants to be a scientist. So the point is that, for instance, Dan Jensen in Santa Rosa in Costa Rica has been doing really great job in forming a school really of experts, of botanists, and all of us that work there, we accept them, and they are basically, even if they don't have a formal degree, they're called para-botanists, and one of them actually has a certificate from a university because his knowledge is compared to a trained botanist to do it, but he didn't do it. He learned it in the field. The other thing is more our experience. Our experience is that it's a situation in which we have the local people are not much interested here in the Yucatan Peninsula to go to university. The one that work with us, their sons, yes. So their sons and daughters goes to university, but they themselves don't. But they want to do ecotourism, and so what we give is information and prayer that they can actually then transfer to their own business where they don't work with us, and so on. So there are various ways, but I agree completely that we need to focus on giving something to that. But my original question, because I didn't know the Sarah's question beforehand, was linked to Katya's comment and your answer about the measure probably is linked to what you want to, the type of measurement is the type of question you want to answer. And I think the example of the group size is a very good one because, of course, nobody really cares that the brain size is linked to group size. Group size is a proxy of something else. But it's only that it's easy, is readily available. And then, of course, both you and Eduardo mentioned that actually is not as easy as we would like to get it. But the entire premises of that, the brain hypothesis is, social brain hypothesis is that there is complexity. And as we know, group size per se is not complexity. So the idea is that if you really want to address that, then you need to get, and then I think it's nicely linking to your idea of description, you need to get more to the gritty thing. You need to get how the individuals in that particular group, with that particular group size, they actually interact. And then those type of measures should be the real one to put in this correlation. But of course, it's difficult, then we know. But if the more people probably will do the descriptive work and the details, then probably is available as well. So I don't know, I would like you to comment on this. It's not a real question, but what do you think about this last point?
Yeah, I think that's a great point. I think that, right, for the social brain hypothesis, group size is a proxy often for social complexity. I think that when we have a primate bias, since a lot of primates form cohesive groups, this is something that we've been talking about in this series. It's like, there's a huge variation in social systems across mammals. We sort of have figured out that a lot of primates seem to form groups, but now maybe with more observations of different species, we're seeing, wait, some species don't fit this pattern. I mean, it takes sometimes years and years to figure out the social system or the social structure of an animal. And then something like group size, are you really comparing the same value across species? And there with social brain, there are many sort of social intelligence hypotheses, right? I think you raise a great point about you need to watch the animals to really see how to capture maybe complex sociality, complex cognition. To a large extent, the brains are also a proxy. It's a proxy for cognition.
Yeah.
And then there's some iterations of these hypotheses that are maybe more focused on social complexity, social cognition, things like that. I think your work on, fission fusion species is really valuable here. And I'm just reading more about the chimpanzees at Gombe lately, maybe because I couldn't get to the field to Ngogo this summer. But, it took Jane Goodall, never figured out this. She's an amazing, amazing observer of chimpanzees. She never figured out their social structure. It took 8 years, 10 years. And it was only from other studies saying, wait a second, I think it's not that they're just going around nomadically, that these subunits are actually, these subgroups are actually part of a wider unit group, a wider community. And then eventually 10 years into that study, they're like, oh yeah, yep, same here. Mm-hmm. So I think that also just emphasizes the importance of description, but also this back and forth between zooming in and zooming out and maybe studying multiple species. I think this session is really great because we're talking to people who study a wide range of mammals too.
Yeah. Yeah, and I'm talking about really about the interactions. You know, you want to see, you have you can be two primate species in which they have a similar group size then on average, or let's say you have the range of similar group size of within species variation, but one, they basically do nothing with one another. They just move around and they are together mainly for predator protection, nothing else. And the other, instead, they need to do a lot more because there's a lot of food competition within, so they form coalition and so on. So, group size at that scale, if you compare those two species, they are basically orange and apples. And that's actually even done congenetic. There is studies on squirrel monkeys that they have a variation, even if they are different species of squirrel monkey. All squirrel monkeys, they actually interact with one another on a consistent basis, rather different depending on the species. So the size of the group is the same, the overall dynamic is similar, the dispersal patterns, but basically, you put the contest competition in it because the resources are distributed in a particular way, bang, and you get the difference. So that's why I think depending on the question, what Kaya was mentioning, I think then you want to have a particular kind of measure. But of course, it's difficult to get it then when you want to get it in a broader sense. But the description will give the possibility to everybody then to dig it. Instead, if you get for everything only the group size, that's basically what you get.
Yeah, that's a great point. The value of description, and primatologists, I think we're in a great position because we do study, again, so many aspects of social life.
Our next question is by Joshua Lapargola.
Hi, everyone. I'm a postdoc with Christy Reel at Princeton University, and I study cooperative breeding in birds. So I have a question that might be a little incendiary for everyone here. I don't know. So in cooperative breeding in birds, there are variations in the proportion or the prevalence of this behavior across birds, though some people have estimated that it's present from 3% to 9% of species. And a lot of the uncertainty comes from the fact that we know very little about species that live and breed in the tropical areas of the world, or most of what we know about birds comes from the temperate zone or northern latitudes, because that's where a lot of people who study birds are. And so there have been attempts to understand the global biogeography, or more broadly understand the drivers of cooperative breeding in birds, and using large data sets that don't really take into account the nuances within different species, especially those that are poorly known. We lose a lot of important information. And when I was doing my dissertation, I came to see these comparative analyses and think these are really cool ways to look at these broad patterns. But at the same time, it kind of sends a mixed signal that we know a lot more than we really do. There are a lot of species for which we don't even know what the nest looks like, let alone what the parental care system is. And so, all right, here's the incendiary part. Should we as a field maybe move away from these super broad comparative analyses? And if we are going to do that and increase our valuation of natural history and just the basic description of parental care system, how can we do that? That's a messy question.
No, I think you're getting to the heart. Maybe I was beating around the bush, with the talk. That's exactly what I'm trying to grapple with. And I think that it's worth, depending on the trait, we may have to do that, depending on what we're interested in studying. For some things, it's wait a second, if there's so much variability within species, or if there just aren't high quality data, then we just cannot, I think it's not worth doing the comparative analysis, because what are these even these amazing comparative analyses that sort of spark our passions and our interests and our curiosities, if they're still just sort of their hypotheses, their stories about what's happened in the evolutionary past. There's even at the best level, they're still just inferring what happened in the past. So maybe we should really, really be careful before we start telling stories about what we think is going on, that we really know the study system, we understand the variability. I mean, to one extent, you may find that there's so much variation within species in a trait that it's not even like it's not even relevant to look at across species. And I think the compromise would be you do the interesting, fun, exciting hypothesis test, like testing these grand hypotheses in a wide range of species. But whenever you do that, you have to be publishing at least a little bit of data, sort of contributing to the cause, to the collective mission of real science. Because I think with the replication crisis, some of these big hypotheses, if they're not replicated, they're not really adding to the science. They're just inspiring. I mean, they may insofar as they inspire ideas, they inspire people to go collect data. I think that's true. But I agree sort of with your impulse, the incendiary comment. I'd also, be happy to hear from others on this specific topic, including, anyone, if does anyone else want to chime in before we go on?
I'm on the list. I swear I wrote down my name to be fair. I'm not, so I am next. And it's great. It's good timing because I was some of the things that Filippo and you discussed, and then Joshua really are a lead to something that I want to say. Actually also connected to what Kaya was bringing up. In trying to bring all those comments together to my opening remarks, I think that's something that we will have to, each of us, decide individually and as a community, that sometimes less is more. We cannot accept by default that more is better. More is better only if the quality is there. And I think that if we're going to do some of these things, we may need really to go into the nitty-gritty details. And for doing that, we will have to be looking more and more, and I'm talking to my fellow primatology colleagues, we will need to look outside primatology. So you open, Aaron, your talk with reference to Crook and Garland. We travel back to the 60s and 70s, and we're doing graphs between group size and where they're nocturnal or diurnal, terrestrial or boreal. We've replaced that by brain volume 5 decades later, because nobody really knows what we mean by brain volume. There are people outside primatology. If they're primatologists, they're kind of neurobiologists doing primatology. So all of these are proxies. Do we want to work on getting better proxies? So you said our discipline is observational. So we're constrained, and there's a lot of power, there's a lot of value in what we do. But shouldn't we be looking outside primatology for trying to speculate on what are better proxies? And with fewer taxa or one population, really start going a little bit into the details. I mean, we keep talking about a home range like they were doing in the 60s. And that applies to every single proxy we use. How many more decades are we going to have of relating the patch size to something else? Or same for the measures of brain, but what are the people studying the brain saying is a proper proxy for social complexity, which is another proxy. We go from group size to social complexity, but social complexity doesn't evolve really. So I just wanted to follow up on all these comments, Kaya, Filippo, yourself, and Joshua. I think that we need it all, but as a discipline, we really have to start looking outside primatology and try provoking some qualitative changes in the quality, in the richness, fewer data that are closer to what we think are the processes that we're really trying to describe. I'd like to hear your thoughts on this.
Yeah, I think that's I agree. I think that's a great way to summarize or synthesize a lot of the thoughts that I've been having. I think what other people are raising. One thing that I also think about, sort of the danger of some of these broad comparative studies that are testing some major hypothesis or major sort of theory in our field, is that also because what we're doing is, again, the human, the observer is sort of the apparatus, to some extent having these hypotheses, these guiding principles of our field, they're good because they send us out to collect more data potentially. But I think there's a risk in that sometimes theories can limit us to only see certain things. Like what I was saying with it, to some animal behavior being a qualitative science to some degree, you need to understand how you go from observing just an animal moving to being able to chop up what are the different behaviors and getting into more complex things like social complexity. So I think to some extent, some of these hypotheses like the socio-ecological model or even things like kin selection, these sort of theories limit us in our ability to see what's actually going on. So I think maybe that's also maybe related to some of the concerns about these broad comparative analyses. That might be a little bit of a tangent. Yeah, there's a lot of ideas floating around.
The next question comes from Karen Stryer. Karen?
Hi, thank you so much for a great talk, Aaron. It was really interesting to hear all that, and some of it was new to me, so I really liked it. I just wanted to say two things that really ask questions about two of the things you said. And they're not really related to each other, but since they were both from your talk. So one of them was about, you mentioned this also in the questioning, in the early questions about looking at variation in group size. And, you know, we did this analysis with Phil's Lee Anthony Ives a couple of years ago about, which showed basically, I think the most interesting result from that paper was that the longer study, the longer term study you have of a population, of the group size in a population, the more variation there was in that group size. And I thought I think that's the most interesting finding of that paper actually, because it really raises the question of when, and maybe questions, do you think that if you study, if you get groups enough years or enough data points from the same group, will it eventually asymptote to tell you what's, you know, sort of what's really going on, because we haven't found it yet in any of the primate data we looked at. And I know this data, people have these data, but people don't usually publish their data. You know, you don't publish a new paper every year when your group size gets bigger or smaller or something happens. So it's really hard it was really hard to find those data in the literature, and oftentimes that was a real constraint. But I just wondered, you know, I guess I think that that's still an interesting question, that studying that variation as pattern, so that no, there may not be something like a species average group size. But I think what we focus on, and I think it's maybe a different question that you were even getting at, is what is the value of looking at variation in variation? That is, is there cases where different species or different social systems promote or prevent or promote variation? And that was a question. And then my other question, so you can answer whichever one you want or address whichever one or both, was again, your idea. I think it's great to think about collecting more natural history data. And you may have seen Shirley Strom wrote a paper about this after her IPS talk in Nairobi too, which is really interesting. But I think maybe, and I know that cultural anthropologists grapple with this too, which is how we can, are there ways that we can standardize the collection of natural history data or these sort of naturalistic observations that is some kind of a compromise, so that on the one hand, we're not so focused that we're missing all the things that you pointed out in that really interesting example. But on the other hand, that we're still collecting the data in a way that can be comparable both across studies and over time and across species and interspecific and so on, because that of course is the sort of arbitrariness of just what I think is interesting may or may not be the same as what you think is interesting. And so we're going to look at different things that ultimately makes it more difficult for comparison. So I guess those are my two questions. I'd love to hear what you think about them. Thanks.
Yeah, thank you for those questions. Those are really great questions. I think, well, first, just thinking about natural history, I think you've exactly articulated what I'm thinking in terms of how we go about this sort of targeted natural history. I think we're now equipped with data collection methods, like collecting maybe data on a digital device. We can sort of collect more data. We can automate some methods so that we don't have to record the date and the time and maybe not even the GPS location. There should be some things that everybody's collecting. And I think that some long-term projects have these protocols in place, and having those discussions, sort of reconnecting and talking across sites and across species, and also studying, if we can, visiting other sites, doing brief studies of other species, that will also help, I think, standardize our methods. But then keeping in mind within that, we still need to be open toward the qualitative aspect of changing, say, the definitions as new, you notice new behaviors or new subtleties, things like that. But I think, right, we don't need to return to an anecdotal phase, from the early 20th century of explorers, reporting on primates, or even the more just natural history that's sort of, you look and you record whatever you can, but it's this targeted collecting certain traits that we're interested in. That's from these broader hypotheses. So I think these big comparative studies are useful for that. And we can see, oh wait, we can compare these species on this, or we're missing data on this, this trait. So I think that's from the natural history perspective. And I think your questions about variation, I am definitely interested in looking at variation as a trait, and somehow potentially comparing species, or just looking within species and what are the predictors of variation. For example, something like group size in the same population over time, what's predicting that? To what extent are things like ecology predicting variation, group size? I think that also sort of relates to this more holistic approach. For example, with the grinding stones in Kibale, this was a section of Kibale that had limited human impact, but there are grinding stones everywhere. This hasn't been incorporated really into formal ecological analyses, but you're seeing in Kibale various patterns in fruit production and various patterns in primate populations. And I think part of that is because it's a dynamic forest system, that in a lot of places of the forest, we're always seeing it as a snapshot. And so over time, through these long-term field studies, you're seeing the forest change, and then the primates are responding to the forest changing. For example, forests growing where there used to be grasslands that were maintained by people, maybe 100 years ago, or for much longer. And this also sort of questions what do we really know about the habitats? What do we know about the forests? But yeah, to what extent that's going on with various primate populations will differ. But I think environments are so variable that, to a large extent, I would guess that it would be hard to find, okay, this is the group size. It's sort of plateaued because we've watched them enough, if so many other factors are changing.
Well, I don't think the group size would plateau because it's not always going up or down. Variation isn't always, it's not unidirectional, but it's that the variation would asymptote.
Yeah. Oh, that's great. Okay. That's
that you have enough time. So it's a sampling question, right. Really.
Yeah. Yeah.
Thinking of it as a sampling, thinking of variation as the sampling question is the unit of analysis, as was asked earlier,
right? As
opposed to the group size per se. It's variation in group size. Anyway, thank you so much. I'm sorry I have to run because I have to go to another meeting, but this was great. Thank you. And I look forward to seeing everyone again soon. Bye.
So we're going to take some questions from our colleagues watching the presentation on YouTube. And then we have Suleima, Susan, and Karsten. Miles?
Hi, everybody. I'm Miles Machinsky. I'm a master's student in Lauren Hayes' lab at UTC, University of Tennessee, Chattanooga. I have some questions, well, 2 comments and 1 question that I'm going to relay from YouTube. First, the easier one, Ben Finkel wants to say, hi, Aaron, and compliment you on the talk. And then David Watts also says, hi, Aaron, and has a comment that goes along with the grindstone conversation. He says that Gary Aronson dug a test pit near one of the grindstones and turned up a 400 to 500 year pottery piece. So that's an interesting thing to think about. And then the question that David Watts also has is, what do we do now with the huge pre-2016 primate comparative analysis literature?
Well, those are all really good comments. So first, with David Watts's last question, what do we do with the primate literature, the comparative analyses themselves? I think there are two strategies I can think of. With the comparative analyses that have been published, this is where we can start doing more meta-analyses, looking at studies that have been published, looking at effect sizes across different studies. But in doing so, we always just looking to what data are they relying on? Because we may find that there are plenty of studies that have been done that rely on high quality data and seem to have a good range of species. So we can feel good about their conclusions more so than maybe others. But I think it's, as we move forward, it's more, I would feel like I wouldn't want to publish a comparative analysis unless I was meeting all these criteria for data quality. And I mean, like I've grappled with this because there's sometimes questions I really want to ask across a wide range of species, and it's hard to do because it involves a huge amount of work. I mean, it's almost like the same amount of work that goes into watching one population to really go through these other studies, and this like a detective that going through an archive, really extracting what's going on in different populations. But I think, yeah, we just got to have a careful eye to these comparative analyses in the past. Future studies will support or refute them to varying degrees, but just keeping that eye to data quality, making sure that in the future we publish the data sets. I think that those are my first thoughts. And then just on David Watts' other comment about the test pit showing maybe a long history of humans in Kibale. Another component is, you know, people were living in Kibale, people were living there like until around 1930s. So that's a very recent history, and that the people are still alive who once lived there. So I think that's another thing to think about. A lot of the tropical forests where we work that sort of seem so remote and so wild in their pristine nature, oftentimes they have a long human history of management. That's just something to keep in mind, especially like thinking back to what we were talking about earlier with supporting staff and scientists in the countries where we're working, acknowledging that that it's not like we're just escaping into this, we're not explorers escaping into pristine nature, that humans live in these environments.
The next question is from Suleima. I read your chatting, Suleima, so you're gonna try, let's see. You need to unmute yourself. Suleima, are you there?
Hi,
Yes.
can you hear me?
We can hear you well, yes.
Oh, goody, okay. So I'm Suleima Tang Martinez, University of Missouri in St. Louis. I'm retired, but still very active. So Aaron, thank you so much for that talk. I found it really interesting, both, or not just both, but on several counts, the issues about methodology, and then also the issues about the importance of more natural history. And then lastly, which is what I really want to comment on, is widening the definition of scientists and who is a scientist. And, you know, I personally have felt for quite a while that when people work in underdeveloped countries, and especially with local people from the countries that they're working in, that some of those people have a sufficiently broad and amazing amount of knowledge, and that they contribute in that way informally. And I think we have to start thinking about making some of these individuals co-authors on our papers, so they get more than just, you know, an acknowledgement mention at the end. And I think in a way that is a way of, that would be a way of changing or of correcting what I consider to be intellectual colonialism. Recently, I was a co-author on a paper on the history of animal behavior in Latin America. And one of the things that was so striking was that modern evolutionary biology really started, and many of the most important ideas were developed in the Southern Hemisphere, in areas of the world that are now developing. And if we think of Darwin, Alfred Russel Wallace, Bates, and even more recently Hamilton, they all spent enormous amounts of time in the tropics in Latin America, for example, but not exclusively Latin America, obviously, and developed many of their ideas there. But then when their papers were published, when their findings were published, they were really directed at an European community. And so little of that actually came back to, or did anything to promote science in the countries where the ideas had first been developed. I also, and I noticed that somebody else, Joseph Berger, wrote about this, but I also think that in terms of widening the group of people who are considered scientists or do scientists, that especially given what the cultural situation, historical situation we're in right now, that it's extremely important to take real measures to increase diversity and inclusion in the sciences. And as I said many, many times before, I don't see it merely as an issue of social justice. I see it as an issue of having more different perspectives, different, people out there doing science who can bring in new perspectives, new interpretations of data, and so on. And I would include, obviously, African Americans, like Joseph said, but I think that all underrepresented groups in science, we have to actively work to incorporate them into the work that we're doing. And again, I think it's to the benefit of science to do that. And then the last thing I want to say, and again, this is something that I've written about, which is somewhat related, although not exactly, is that so many, because of the imbalance that has existed in terms of where science is done and who is doing the science, there are so many theories, hypotheses, models that people have come up with that are based largely on species which are Northern Hemisphere species. And I'm thinking of rodents, for example, where people come up with ideas on the evolution of sociality, but up until relatively recently, those had been based primarily on Northern species of rodents. And I have argued very strongly that, for example, caviomorphs, which are almost endemic except for the porcupine to Latin America, that we really can't come up with broad general theories or hypotheses about various aspects of animal behavior or biology if we don't take into account the large numbers of species that are in tropical areas that may not behave at all like the species in the north. And another, I mentioned rodents, but another example is birdsong, where in the Northern Hemisphere, mostly males are the ones who do the singing, but in tropical regions, you have males and females singing equally. And oftentimes we forget that female song is important and needs to be studied. So that's what I have to say, and would love to hear, Aaron, what any comments you may have on that. Thanks a lot.
Thank you for those comments. It's hard to even follow. You raised so many, I think, points that really resonate with me, and I think I just sort of agree with a lot of them. I think your point about widening who who's a scientist in our field. So forgetting, I mean, we want to do it in places where we're studying animals in the wild, but also we want to make sure that we're removing any obstacles that seem to be limiting people from joining primatology or animal behavior. I think that's a really great point. And having a diverse array of scientists will, I mean, it's so important for animal behavior. Like it is important for social justice. The fact that I think that primatologists, animal behavior, we all have to be anti-racist just living in this context and take that seriously. But since this is behavior, we're just trying to get into the mind of another animal. We need people from as many different perspectives as possible trying to do that, and who have different experiences and who have different cultural background, who have these different lenses or biases, because each bias that we have is a sort of a window into the mind of another animal. We have our own theories. So that's again where there's a back and forth, but the power of theory to make descriptive observations. I think yeah, you sort of I think did a great job of identifying so many important issues as we move forward in animal behavior. So thank you for that. Eduardo, you're muted.
Once again, thank you, Suleima. And the next question is from Susan Perry.
Hi, thanks for the talk. There are just so many points you made that I'd love to comment on. Obviously, I can't comment on all of them due to time restrictions. So I guess maybe what would be most constructive is if I talk about the challenge of operationalizing natural history data collection reliably. And so this is something I deal with a lot since my long-term study, which is, for those of you who don't know me, is a 30-year study of capuchins. I'm mainly focusing on behavioral variation and trying to understand patterns of innovation and cultural transmission across many behavioral domains. And so I have a ridiculously elaborate data collection protocol with hundreds of behaviors and, for example, combinations of objects, physical actions applied to them, and of course the IDs of all monkeys who are interacting in every single vocalization and gesture they do. And so I find, and I run my study by having volunteers who stay on average 1 year and rotate in and out. And so I can't really emphasize enough how important it is that me as the PI be there at least 4 months out of the year, scattered throughout the year, to make sure there's no drift in data collection. So long-term managers help a lot. Unfortunately, I can't always have one. And so what we do is we have not only very stringent training exams, but then every month we re-administer those exams to make sure there's no drift. And even so, there are always going to be cohort effects, for example, some cultural drift in how things are done, and that can't be prevented, but at least you can tag every single line of data, not only as it's collected, but when it goes into the database, to say who had what role in the construction of that line of data. And that can help in, address a lot of these problems in data analysis later on. And I guess it gets to another comment. I'm very enthusiastic about the open science movement, but also very wary, from past experience, knowing how easy it is to misinterpret somebody's data, particularly when it's a very complicated data set. And so I would like to advocate for the very active involvement of the data collectors in any kind of subsequent use of those data. I mean, you can make a lot of metadata, but it's impossible to anticipate all of the misunderstandings that are going to happen of the use of data sets, especially when they're used for purposes other than the one for which it's collected. So yeah, I guess I would be interested in, as this talk series unfolds, in knowing a little more about people's data collection and data storage processes. I think that we probably can all learn a lot from each other in that aspect.
Yeah, thank you for, those are, again, really great points. One comment just I have, I really like the fact that you are emphasizing that the data collector should be part of the analysis, just because that you need that to sort of make sense of the data. And that sort of goes back, reading some of the literature on qualitative methods, people make similar arguments. If you're doing with qualitative data, the one who has collected the data sort of needs to be the one to do the analysis to really make sense of it. And I think if you have clear definitions, like you're emphasizing, Susan, the inter-observer reliability and the training that goes on, that's so important. I think that's something also that should be more emphasized. I think in the talking across sites, I think would be really valuable. We do more of this collective effort. And then Eduardo is muted.
Thank you, Susan. The next question is from Karsten.
Yes, hello, I'm Carsten Schradin, based at the CNRS in Strasbourg, France, working on striped mice in South Africa. Thanks, Aaron, for a great and inspiring talk. And when I put in my question mark, I wanted to make a comment, which you in the discussion just made 2 minutes later, and Eduardo made it again. But for peace of my own mind, I would like to repeat it. When many people here say that descriptive natural life history data are so important, of course, we don't mean that theory testing is not important. I think theory testing in case studies and comparative studies, this is what made our field big, what made it important, what gives it meaning. But the quality both of case studies and comparative studies depends on the life history data. And all of us who work in the field, we do collect these data and they should be published. Now, the problem is publishing these data doesn't give you any reward. And we have to remember that most of us in behavioral ecology, we don't have permanent positions. I remember when I was in Zurich without a permanent position, I had as a tactic, a strategy written down on the wall, don't spend time and energy in publishing papers in low-ranking journals because it won't help me getting a job. So from this point of view, I think it is the people that have permanent positions that should invest into publishing these valuable data which they have. And I think we all know that these data are valuable because it takes a lot of time and energy to get them and to publish them, even if we don't get them into high-ranking journals. And the other point I always would like to make, also to invest into studying species. And now I say something which I've been taught for all my life we shouldn't do, to make a study because we don't know much about this species, which is a phrase that is like a no-go. But in the meantime, I think for comparative studies, we must exactly do this to some extent, like having, for example, some master students in a row to work on this. Then the main problem, and this is what Aaron mentioned, are the quality of the existing databases, because it is very nice to take these databases, make a lot of sophisticated analysis and get a great paper, get a career. But if this study, for example, that was published by Eduardo and Corto showing that the data in databases like Pantheria is highly unreliable, then we have a big problem. And at some stage, we might have to decide to get away with these databases and build up new high quality databases. But then the question again is, how do we control this? I mean, we need somebody to review the quality of these databases, just replacing them because, okay, now I come and now I have the much better database. Is this true? I mean, this must also be somehow checked. And I think if we look at behavioral ecology in the mid to long term, I mean, the next few decades, we must be on a way to create better, higher quality databases by publishing this life history data, but I do not know how we would be able to review the quality of something like this because nobody will get rewarded for this or get a position for that.
Those are, yeah, thank you for those points, or synthesizing various themes that you've been talking about. We've been talking about the series and talking about today. One thing that brings to mind is sort of how we go about publishing in general. I mean, to a large extent, like it's we, the scientists, are the ones that it's this collective enterprise that we're doing. And I think we might lose sight of the power that we have if we gather together. And I think that, for example, Karsten, you, Eduardo, others have been able to move the field in a certain direction, getting journals to have certain sort of guidelines, things like that. It's one step in the right direction. Another thing that also makes me think of maybe we should just be valuing publishing data sets. And there's a trend towards being more transparent with your data, your analyses, journals that allow you to publish just data sets more rather than even having sort of the hypothesis. But that also makes me think of just another problem that is sort of related to this, which is the corporate nature of publishing. And that this is just another thing where we just need to, I mean, this is a great start. We can gather together, and hopefully no one from like Elsevier or Springer is listening, but like we need to rethink this whole way that we're publishing and who's benefiting from that and who's bearing the costs from that. Because I think it's also really problematic when we talk about access and talk about diversifying the field and making it more accessible to people in other countries, including the people we work with, that there are these huge paywalls and that there are these various journals. So I think we can sort of we can maybe as a group expand and working together, having this collective action to shift really just what's valued. If we just start publishing, I mean, that's how journals got started, is people just publish stuff. Primatology especially is such a young field in the grand scheme of things, that we can just set the trends, and hopefully the field will follow.
Thank you, Aaron. And I wrote my name again. So yes, and I definitely want, before you said, before you made a reference to the business of publishing, I had made the note that that's what I wanted to follow up. So really my comment is, thank you for that comment. I was thinking if we should, I would like to invite us to really realize that we will be responsible for the demise of our small journals in our disciplines unless we do something to counter that tendency. It's like we can think of our journal as the mom and pop store. And either you go to Walmart or you purchase from the big companies, or you keep making valuable our journals. And the tendency, what is high impact? I mean, you're talking about Springer, I mean, Wiley. We had a big back and forth of messages because the impact factor of AJP had increased in the second decimal. And I bet you that most of us could not even produce a paragraph describing what it means. Who cares? But unless we defend our discipline journals, they're being swallowed. I mean, AJP, all these small journals, the only mandatory thing in the author guidelines for AJP is the graphical abstract. Do you really think that that was our idea? Or it's the press who wants to sell this like a visible product for someone to consume? So I would like to think that it is our responsibility. By that I mean those who already have, like Karsten said, established position. Let's see if out of this, at the end of the semester, we've already been talking to some of the people in the audience who are editors and associate editors. Can we identify a small set of journals and a small set of us who make a commitment for a number of years to, we don't care about high impact anymore. We're going to go to the disciplines or to the journals that we think we have room and possibilities of changing things. Because who decided that we can only review a paper in 14 days? They're not asking us to review papers based on the quality. Important is that you get it done fast. Why? Is somebody's going to steal my findings about Almacis in Formosa? So we're being swallowed by fields that have nothing to do with our ethos. And so if we care about our ethos, we will have to put our actions where I'm putting my words. I'm willing to do that. I invite people with this. This can be another conversation. We have pending the one about methods. And the last comment is about what we do, but this is for David. What we do with studies published before 2016. What we do with any study. We look at the methods. We look at it as different from a report in the Sunday newspaper. We assess individually as trained scientists whether we believe in the quality of the paper, and then we decide if we use it or not. That it's published, that's what we do with the paper before 16, before 14, or before 2019. We just evaluate it and make our call. That's all I have. If you don't have any comments. So don't worry, don't worry about Springer. So we're behind you. I mean, we need to defend our disciplines and the quality and the rigor of the work we do.
Yeah, and I do. The one other thing that I maybe add is especially what Carsten was bringing. How do we control these data sets? I mean, that's another important question. But I think if we rethink how we're publishing, what's valued, I mean, I think we need to shift to this more collective, where a journal could be a database, and you could be publishing, rather than publishing a data set, the journal itself is a database, and someone managing that. It's like, what we don't need some corporation profiting off of our research that we're not getting anything from. If they're gonna be profiting, let them be managing a database, something like that.
We have 5 more minutes. I have a question from Rachel, and then after Rachel, there's Filippo. And after that, I suggest that we officially finish the Q&A session. I do have another meeting at 1, so I can make somebody else a co-host if people want to continue talking. So Rachel Boyd, you're next. And after Rachel, again, I said, if we have time, Filippo. If not, I'll make somebody else a co-host. Rachel?
If you'd like, we can skip me because I'm.
No, please, no, absolutely not. You're next.
Okay. Well, hi everyone. I'm Rachel. As I said, I'm one of Aaron's students as well as Tony's students, and I actually really love the conversation on publishing and what we value. But I want to go back to thinking about theory in primatology and other studies, and kind of in the same way that Joshua a while back was talking about or questioning the worthwhileness of doing comparative studies, and also like adding on to what Zulema was saying about who were involving, and also what Susan was saying, and others like who were involving in research. I was just wondering what you, Aaron, thought, and I know we've kind of talked about it, but also what others thought about the worthwhileness also of testing theories or continuing to test theories of animal behavior, given the biases in who's been allowed to produce that knowledge, whose theories have actually made it into the canon, and like which species these theories are originally based upon, not to mention all the issues in data quality. And I know that Carsten was saying, like, yes, we should still be studying theory, but given all of these limitations, I didn't know if it was worth it to keep testing them, you know, before we actually like get our data quality in order.
Yeah, I think that's a great question. I think we should only test them if we have our data quality in order. But I think we could still test hypotheses or these sort of theories. But when we do it, take the real time to do only include high quality data. And I think part of it, it's like, Rachel, you're saying, you know, some of these hypotheses, what are they really based in? There's all these biases going into them. From my perspective, to a large extent, we still want to have fun. I mean, these are just animals, you know, these are primates. They're interesting to us. So we should sort of be testing hypotheses that interest us, testing theories that interest us. If there's a theory that you think is wrong, going and then trying to refute it with high quality data could be a lot of fun and useful and beneficial to the field. But also I think that that's why the spending time observing animals, returning to this descriptive phase, you may come up with a completely new approach or a new hypothesis. And so you don't need to worry about testing some outdated thing. Although I think, right, sometimes in a grant or an article, they're saying, wow, why aren't you citing Bateman's principle, something like that. But then you could cite people who, like Zulema, who've critiqued that and others. So I think, right, striking the balance. And not everyone, I think, also needs to be doing comparative analyses. I think they're the two aims of primatology. One is the comparisons across species to really infer the evolutionary processes. But then the other one is just these really detailed studies of primate populations. There's plenty of research to still do just on one study population. And then I think that you could just do that. I mean, that's still primatology. That's really useful. That's still animal behavior. You could spend your whole career just doing this really detailed study of that population that will benefit others who want to do comparative studies. I would just say that I think it's a little risky to only do comparative studies if you're not also grounded in a detailed descriptive study, because that's where you're not going to be able to filter out what's maybe a bias or what's not actually interesting or what's not relevant to the species.
I think that I prefer to stop now. Some of us have meetings at 1, and Aaron has, for those of you who don't know, he has individual meetings with many students for another 2 hours. So it may also be fair that we give Aaron a little bit of a break between 1 and 1:30. Before we officially finish the meeting, I'd like to remind you that next week, Michael Taborski from the University of Bern in Switzerland will be sharing a presentation titled Shell-Brooding Cyclids as Evolutionary Models for Intersexual and Intersexual Conflict. Did I say well? Intra and intersexual conflict. Michael Taborski next Tuesday. So thank you so much. And we look forward to seeing you all on Tuesday. Thank you so much, Aaron. I'm impressed. I really mean it. Thank you so much. We have still 30, 40 people joining us for a full hour of questions and answers, provocative questions. I don't think incendiary, just thoughtful questions. So I thank you for that, and I look forward to seeing you all on Tuesday. Thanks.
Thank you all.